my great pleasure to welcome you here for the Insitute-wide Kolloquium at the IPHT, and especially welcome our guest today, Manu Prakash, who went all the way from Stanford first to Dresden, now luckily also to Jena. And so if you don't know him, he's very much famous for also the full scope, but especially for discovering the wonders of nature by following frugal and curiosity ideas in a way. And so I've met Manu at multiple occasions, and we also spent some time in the lab in Stanford, built some of these cube-based microscopes just to advertise them once more. This year was also part of some projects where we wanted to try imaging marine samples in 3D, and that's also something where we're still collaborating with. And yeah, I don't want to talk too much about these kind of things, so stage is yours. I would recommend just questions during the talk, afterwards.
In the talk, interrupt me anytime.
Yeah.
So thank you so much.
Thank you.
This will be very informal. I can talk about hundreds of different things depending on people's interests. So just so I know, there are people from the Institute, the Photonics Institute, but are there other folks from other places as well, just so I know? From
also Leibniz-IPHT on aging.
On aging?
Chemical ecology.
Chemical ecology. Anybody else from zoology or from the Institute of Zoology? Okay. Yeah, I think usually I don't have any particular agenda other than I was going to touch upon a few sets of things, but also just more philosophical ideas itself. It's rare to have an opportunity to spend time with people. Even in the last day or two, just the history of this place reminds us that both the capacity to invent new instruments, but apply them in new places just leads to wonders. And, you know, what I'm talking about is this, personally to me, this juxtaposition of course, Zeiss and Abe being together, but at that same time, Haeckel being here at the same place and at the same time, leading to just remarkable discoveries that happen. I think, you know, often enough in science, we have to be at the right place at the right moment, asking the right question with the right people. And so some of the things that I'll talk about will try reflecting that. I have two options for people to take briefly. I could take you out to the ocean for a little bit to tell you the extreme environments in which some of the optical instruments reveal completely new insights that we haven't had. Especially, we are moving towards trying to understand the role of cell biology in climate change and why and how optical instruments will play a very important role in that space. Or I could start by just sharing much more of the philosophy of sharing scientific tools broadly with people. I think I will cover a little bit of both. I'm just curious, where would you like to start? I think the idea would be is we could start with biological questions first and then broaden the sets of tools, or the other way around. Any votes? Who wants to go to the ocean, and who wants to go to like a trip around the world exploring frugal tools with new applications? Where? Ocean or? Okay, ocean? Yeah. Let me take you guys to the ocean for maybe like 10, 15 minutes, and then we'll kind of return back. But I think it'll become valuable, the philosophical framework of how we think about these types of ideas, especially in the sense of how we discover the kinds of questions, for example. So let's spend a few minutes to the ocean. So as we speak, at this very moment, I have 6 of my students in the Arctic. They are on this boat. It's called Kolloquium. It's in the Arctic. And one of the things that I started doing almost maybe around 8 or so years ago, of course, worried about the climate as all of us, but thinking about how can our science influence and allow us to ask questions, we just started spending time in the ocean. The first realization I had at that time was these boats cost $50,000, $60,000 per day. And being on a boat like this, I learned that there was not a single microscope on one of these boats. often enough, people tried taking stuff out and it breaks. And one of the things that started happening is I started realizing that there's a large class of questions that could be addressed if we were really paying a much more microscopic view. So I'll share with you a little bit of a microscopic view of the ocean. So first of all, why should we really care? Here is the challenge that we have. If you dive, I don't know if any of you dive, you dive into any part of the ocean and you would see this is the density of life that we're talking about. Incredible amount of biomass. This happens to be a night dive. And if you're trying to understand an ecosystem that is really deeply coupled to our climate, this is a very difficult challenge. The number of species explode. If you really think about where we are in the era, of course, Haeckel discovered his very first love was radiolarians. He discovered so many species of radiolarians travel around. Now we do it the other way. We sequence the hell out of everything. So 100 or so million genes have been discovered in the ocean. 50% of them have completely unknown homology to anything that you would find on terrestrial environments or anything we have known. The other part of the challenge, and I'll describe this, is how this life connects back to carbon fixation in a minute. But the other reason we started thinking about the ocean is it's a very strange ecosystem to begin with. It's a one-dimensional ecosystem in the sense of function of depth. So all life and majority of the sets of cells, for example, can have cytoplasmic density that's a little bit heavier than seawater. So many of you know or have heard this term that half of the oxygen that you breathe comes from the ocean by algae and diatoms producing this oxygen. But it turns out diatoms are actually heavier than seawater, and nobody has known whether diatoms don't have any flagella or any cilia. So if they're heavier than seawater, millions of years have passed, you would assume they would just sink to the bottom. And light, for example, in the ocean is only in, say, the first 150, 200 meters. And of course, there is a decay. And after the euphotic zone, it gets very dark. And after, say, 300, 400 meters, you cannot sustain photosynthetic life. But many of the nutrients are actually down there. So it's a fairly complex system. Of course, temperature is varying along that z-axis. Salinity is varying. All of these parameters that you might care about. And so one of the views that we don't have of life in the ocean is along this axis. What we know so far is we collect stuff. We can say a little bit about who was where because you can do collections at certain depths. We have a lot of chemical view of the ocean in that sense. But one of the desires that we had at that time is could we dive with a single cell and really try to recapture, what is the life of a cell, for example, in this open water? And then in the context of thinking, this is sort of what led to the idea that I'll talk about. But in terms of just looking back at a global view, what we do know is the satellite data. So this happens to be the satellite data of the planet, very much like seasons. Every one of these greens is the chlorophyll. So we know massive abundance of blooms in terms of biomass. And often this is a surface view because satellites don't penetrate further down. And what we would like to know and understand is couple the Z dimension to this dynamics. So when a bloom disappears here in our IPCC models, we assume that that carbon cells died, they sink to the bottom, and that biomass is actually the significant portion. It's said that between 40 to 50% of every carbon molecule that we are emitting out in the atmosphere is being fixed. So it's the world's largest CDR technology, the carbon capture technology, but we don't understand how it works. And so that was one of the key ideas. And of course, if you look at mountains of Dover, that's the proof that carbon sequestration works this way. This mountain, I don't know how many, I haven't seen it, but I'm wondering if anybody's actually seen it. It's not too far from here. The mountain is actually made of single cells that are only 5 to 10 microns. The entire mountain is literally made of coccolithophores. And so this is millions of years of sedimentation. So you can see the kind of mass that many of these systems store. So the challenge has always been in this type of a framework so I'll just give you a sense of what we do in terms of trying to do measurements. This is what life at sea actually looks like. I will talk about this instrument a little bit. You can see a whole bunch of wires up there, but it's kind of fun to just see how do you do precision experiments at sea when the problem is, this is what the ocean looks like. So that's the Great Passage. So you're talking about massive waves and often enough, the boat itself, the boat is rocking. So most instruments that we build have gimbals. So it's trying to cancel the rocking and rolling of the boat itself. And then the instrument that I'll mention is kind of the history of the idea of that instrument started with an observation that we made in Madagascar first, which is we had been studying a parasite for a while, and this parasite is schistosoma. It infects humans. So if any of you go in a lake in several continents, there is a parasite that swims and finds humans, and it will penetrate your skin. The unfortunate part is that the parasite travels 50 meters. And we can find the snails that has the parasite, but how do you know where the parasite would be and what is the behavior of the parasite? So we started building just very tall microscopes. And this is one example of a 1 meter tall microscope. We would build these tubes, we would fill them with parasite, and we would try to track linearly where this parasite goes electronically. And you can imagine the problem here that this doesn't scale. Because 50 meters, I asked my department chair, could I please use the side of the building to make this water column? They were not very happy with it. So we had to come up with something else. And we came up with something very simple, and I believe which is elegant, which is this idea of can we do tracking in one dimension to infinite distances, kilometers, and this is what we call scale-free vertical tracking microscopy. And the idea is very, very simple. I don't know how many of you have gerbils at home. It's basically, how can we do something like this, but for a single cell? And the instrument, I'm not going to talk too much about the instrument, but that's what the instrument looks like. And it's basically a wheel. It's a glass wheel, which is quite big. Maybe it's the size of your big dinner plate. And there are two locations in this where we can get hydrodynamic levitation. So if a cell, even a 5-micron cell, is swimming upwards, we actually spin the wheel down. If it's swimming down, we spin the wheel up, and we do 3D tracking in this to be able to get essentially a track of an organism that's here. So in the lab frame of reference, it doesn't go anywhere, but in the frame of reference of the animal, it's going as if it's in open waters. And the reason I wanted to mention this is because, of course, Haeckel discovered many of the radiolarians. And the beauty of many of these cells in the ocean, and I'll show you one example of this for a second, because I forgot to add that picture is an important picture to show you all. He made some of these drawings that I'm sure many of you either accidentally or otherwise have stumbled upon. But one of the important aspects, when you look at these sets of cells, just incredible diversity of shapes and forms of these cells in the ocean. We cannot associate the functional context. Nobody can tell why you need a crown built like this. And I think one of the joke that I have here is because Haeckel and all of us have been doing microscopy in the XY plane, you cannot actually tell whether Haeckel should have drawn that or that. And I am referring to the idea that cells can sense gravity. And out of 500 or so species that we have mapped in this type of a framework, we actually find every single species detects the gravity vector and aligns to it. And so, in fact, I do know the answer of what Haeckel should have done. I'm not going to reveal it yet. I want you to think a little bit about it. And it's very important to think about when we're thinking about cell biology to bring ecology to the table. And this was the big idea of what we are trying to do, is to build ecology into our instruments. So let me just quickly show you some of this data. And then I'm going to show you just one, what I believe is just an incredible data set that tells you why these things are surprising. So now this is the instrument. You can see it's locked in. We built everything from scratch, all the electronics, all the separate components. Of course, there's conventional objectives. In this version, we're doing 3D tracking, and this is all happening at sea, so you can actually see that there is a storm coming. We had to shut down a couple hours after the video was taken because it was just too wild to actually continue. The boat turned around. But what does that what does that data actually look like? So let me show you the instrument and then just show some data sets. So first of all, the instruments are inside temperature-controlled environments. And what I said, an ocean on a tabletop, not only do we control light and temperature, we actually control pressure in these chambers. We can control salinity. So you should think of it as a virtual reality glasses, but for single cells. So if the cell goes 1 meter or 10 meters down, we will increase the pressure in the chamber by 1 atmosphere, which any of you go diving, you know, every 10 meters, the pressure increases. We can change the light curve. We know the light curve and the wavelengths at the same function of time. So the idea is for the cell to perceive as if it's actually in the ocean, traversing these long distances. And then this is what these data sets look like. So for example, I don't know if anybody recognizes this object. This is something that you have all touched and played as a child, but it's the embryonic or the larval forms. This is a bat star. So bat stars have these star-like structures, but when they are in their larval forms, they form these incredible larvae. And for months, these larvae explore the ocean. But we have no idea. So if you think about conservation of species, we have no idea how this thing is making decisions. And this is a small data set of a larva that's actually climbing 0.5 meter. And I'll show you kind of the very fine behaviors that we can measure. In this system, we can actually do calcium imaging. So we can even watch what the larva is thinking in terms of starting to make these class of decisions. And of course, as you notice, it appears as if the object is just floating in front of you, but in fact, it is climbing, and we are compensating electronically to make sure that the object remains at the same place. There are no walls in this, and so you might wonder, oh, why wasn't the larva going sideways? And this is what I meant, that this is not a confinement effect. The fact that this trajectory is ballistic along the z-axis is just what nature has evolved, because it cares about gravity. And even if you give it all the space in the world, it decides to go up and down. And the idea is actually very simple, because the shear velocities in the ocean are incredibly fast. The z turbulence is very, very, very small. So think of it as an elevator. If you want to disperse, all you need to care about is the z-axis, because the ocean will take care of the xy axis. No matter if you swim in this dimension, it doesn't matter, because the currents will take you. But if you swim in this dimension, you will couple to turbulence at different scales. So this is a very fundamental idea. And I think we were surprised to see that even for 500 species data set, everything cares about the z-axis. Okay, so now I'm just going to rapidly just show you some really fun movies. So first of all, just to give you an idea that what you saw, there are hidden degrees of freedom. So now I've tethered and I have lit up the fluid, so you can actually see what a complex, the complexity of the flow field that these sets of systems are generating to be able to do what the larva was doing. In fact, so for example, to be balancing itself in the gravity axis, it's using many of these vortices, it's turning these defects on and off. So there's a very complex controller that's actually built in. And then this controller then gives this type of data set. So we'll just watch this larva and you will see something funny happens at around 150 seconds. The larva is just climbing. You can see that track, it's going up, and right there, we call this ciliary reversal. It did a dive, it did a reorientation, it found gravity back again, and then it starts climbing it. So this is the very first time we can actually collect data sets like these to know that there is some innate behavior in many of these organisms. And so you can see every 150 or so seconds, and possibly maybe I'll slow it down here. Right there, that's the reversal. It does this turn, it moves around, and then it climbs up back again. And we know that this happens to actually be a feeding current. So we have done this for a very large number of species. And one of the big things that we are excited about is very much in the spirit of Haeckel, release a behavioral data set. So very much like he mapped what the shapes look like, we now have the chance to actually say, what do they actually do? So just imagine his book, opening up a book and touching any of those organisms, and then they come out alive, but they're doing an ecological behavior associated with it. And then maybe I'm just going to show you one video to surprise you. that life is actually quite puzzling in the ocean and we don't understand everything. Remember, I mentioned about diatoms. If it was true that their density is actually higher, that means they should sink to the bottom of the ocean, and you shouldn't have diatoms to begin with. I mean, this is paradoxical. We've had diatoms for millions of years, but they are heavier than water. They have glass, quite literally. So how is that possible? And, you know, often enough, an object that's, say, for a given mass and a given radius, you can write down its sedimentation density. And so we decided to test this, and we started putting diatoms. And this movie is the most puzzling movie I have ever recorded in my, I've recorded a lot of data sets. What you're about to watch, it's as if, I'm not going to do this, but if I drop my phone from here, and a few seconds later, it just suspended in thin air. So that's what you're going to actually see. An object made out of glass that's heavier, sinking, and 100 milliseconds, it's now suddenly become buoyant. There are no interactions, it's not touching anything. It actually has the capacity to change its density by this much, 5 to 10% in 100, and then it says, oh, I think it's time to go. So objects that we had thought that are non-motile actually have capacity to move. And they can not only, and we collect these data sets out in the field. So this is a data set that's collected on a ship. When we bring these organisms back to the lab, 5, 6 days later, they lose this capacity. They don't do this because they realize I have abundant food. I don't need to navigate this ocean. And so often enough, these types of phenomena cannot even be seen unless you take them out. We actually believe there's ion channels, really fast ion channels involved in this process. So when many of you think about origin of the neuron, which remains a big mystery, somebody around the same time when we posted this data actually did a patch clamp experiment on a diatom, and they discovered that it actually spikes. So another big puzzle and a mystery. Yes?
So we didn't know, right? That's them last, right? And they showed the same behavior, but then 5 days afterwards they stopped. Or was it like 5 or 6 days afterwards? Might have been them tested and they didn't show the behavior.
No, I think the problem is basically the way we culture them. So we culture them in a uniform media. If we keep them in the samples and environment that we collected at the depth of the ocean, the behavior sort of continues. But over long periods of time, we do bring samples from the ocean back to the lab. And what I'm trying to say, when you have a cultured cell, it's actually, you should expect plasticity in their behavior. And often enough, you know, it's often I work on a cell type and I don't even know when it was isolated, for example. There's big differences, especially if you're thinking about behavior. I think the cell has everything around it. It doesn't need that much to think about. And then maybe I'll just say one word about, I said something about climate change. So this is an important optics problem that we are working on currently, which is really how does the ocean then actually remove carbon from our atmosphere? So what is known so far is that CO2 from the atmosphere gets dissolved. Life in the ocean takes that up to build biomass. And so you get cells. Now cells die or they have infections. We've been studying a role of viral infections. Then they stick to each other and they make larger clumps. And this is what one of those clumps looks like. And I'll show you some of the data to show why just watching is important. And the very first time, I mean, because this Titanic accident happened, many of you probably know the bathysphere. The very first time a human being went down, and what William Beebe described from the bathysphere, seeing was not some incredible life. The first thing that he described, he said, when I think of the floor of the deep sea, I see always a steady, unrelenting downward drift of material from the above, flake by flake, layer upon layer, the most stupendous snowfall the earth has ever seen. And it's called marine snow because this is what fixes carbon. It's these cells that are clumping together. And one of the challenges has been is when you look at much of the data that's been collected on sediment traps when we go out at sea, it doesn't match with what we know the amount of carbon that's being fixed. So what we decided to do is just watch the process. And you're going to watch the very first video of these particles just sinking. Because of course, you would love to be able to do this in situ. We are trying this now to make some of the instruments that we hang on a wire that goes deep down, and that's a difficult experiment. So we put them in the gravity machine, and you can see these objects are all kinds of shapes and forms. And I'll just let you watch just what is actually happening in the ocean. This little thing is literally saving our planet currently, because if this mechanism of sinking was to stop, the climate CO2 will go from 400 parts per million to 600 parts per million overnight. So it's a very important part, and we have no idea in terms of the mechanisms of what is the rate. So the analogy that I would give is, if some of you think about rainfall, rainfall begins with the bacteria, there is coalescence of droplets, there is a critical size, and that's what determines how much rain you will get at a given point. We have good physical models of that. I think the problem here is we don't have biological models of this process. And one surprise, which I think is as a scientific result, I'm quite excited about, but as a human on this planet, I'm quite depressed at this time, which is what we ended up discovering just from the optics. If you notice, there is something very subtle. This doesn't add up. The mass of water that's right behind is actually moving with the particle. So if any of you have taken a classic fluid mechanics class, you would expect the flow field to turn around just like here, and then go around and go like that. But that's not true. Something is traveling, and it turns out this is actually the mucus. You can see the signature of it in the flow field, that giant yellow ball. And what this is doing is the cells are secreting these polysaccharides, these polymers, and they are essentially like a little thread that's holding, and this is reducing the sinking velocity more than 50%. And so, in fact, our estimates of carbon flux in the ocean are off by more than 50%, because nobody actually measured. What we decided to do is measure the size of the particle, apply a classic Stokes law. We didn't realize that there was an invisible material that changes the sedimentation dynamics. So this sort of gets me excited that we saw something that's completely new, but makes me very sad, because in the end, we are overestimating, and it might be that if these sets of cells produce more mucus, many a times we see these particles to be completely buoyant, where they stop sinking at all. So this is all I want to say about the ocean, unless there are questions. Some of this is published, and one of the threads to think a little bit about is sometimes we see very surprising cells. Maybe I'll just show you a video of a cell that we collected from 400 meters, which then has a capacity to inflate. And this ended up being a really strange geometry. Nobody's ever seen this before, that a cell can inflate like a balloon, for example. And of course, one of the things is when we do light sheet imaging on something like this, I'll just show you that video. The topology of this cell, unlike a traditional cell, when you think about that a cell is a ball of cytoplasm, this happens to be N genus torus. So the cytoplasm is not even a single ball. It's this incredible reticulated network. And this topology is what allows it to inflate and deflate. And it's using this inflation and deflation to go up and down. So lots of life forms have evolved to really control their position in the ocean. All the mechanisms are completely new, but we can only discover them when we actually go out there and measure and map at as much optical detail as possible for many of these instruments. So that's all I want to say about the ocean. If there are any questions, and then I'm going to jump to a little bit of the broader framework of just solving problems with communities, and how do we make sure that we are not the only ones that get the chance to sort of play in this playground of science, but a broader group get to play. Yes.
I have a question for you.
Yes.
Like how deep you can go with this your system?
Yeah, so currently we can go 400 meters. And if one of you knows a glass that's stronger, this is a 2 millimeter glass, and unfortunately our microscopes explode at 40 atmosphere. So that is equal, and I want to show you a picture of failures, not just success. So here is a failure. It completely explodes after 40 atmosphere. So in our virtual environment, we can go to 400 meters. And the other instruments that we are hanging down in the water, they are only 100 meters or so, because you have to do a lot of work to make sure that they are both watertight and pressure tight.
So you pressurize only the chamber or like the whole instrument?
No, just the chamber. That's what the cell. So what we do is we send what is called a CTD down. We collect the water, we keep it at that same pressure. We bring that up, and then we have a mechanism to transfer it to this pressurized chamber. What we are trying to do is maintain as many ecological parameters in the microscope as possible.
And then second question, when you looked at this 400 meters down there,
Yes.
and there's no light, so which lights do you use to see the species?
Everything we do is in IR. So all the data I forgot to mention, you see it's grainy and stuff. Everything is collected in IR because we don't want to perturb their own behavior as a function of light. And one of the things that we of course have is there is another panel which is not visible here. There is a panel, right? This is just for testing, but there is a panel up there that emulates light at a known depth in the water, because wavelengths have a very different distribution. And one of the fun things we can do, which is going back to virtual reality, we can put an environment that does not exist on our planet. For example, when it's going down, I can crank up the light. No organism will ever see, as it's going down, more light. And what we're trying to do is mess up with their circuits to ask, what would you do? Like if you look outside and it's very shady, you think it's a very it's going to rain, but it's extremely hot. What would you do? And it's actually very interesting to think about. All organisms take many confusing cues and build a version of the world, but we now have the chance to kind of crosswire to be able to ask what cues are important. And this is why kind of the direction for all microscopes that we build, we are trying to incorporate as much ecological parameters as possible. Yes.
Asking question. I heard about this diatom that there are actually photonic crystals.
Yes.
Of the light.
Yeah.
It's kind of floating for
Yeah. Yeah, I think the comment that I'll make on that is it's still an open question. People have tried and shown. Frustals, this is why diatoms have glass to begin with. They do have these very beautiful photonic crystal-like structures. People have shown that it has UV protection. So that is very well established. It is also well established that glass is difficult to crunch. Copepods chew on it, but actually the copepods have now co-evolved very strong teeth. So it's very clear that diatoms get eaten all the time. And there might be other mysterious functions because they have to do a lot of work to remain in the water because they are making glass, which is heavier. And they are actually some of the fastest sinkers because of that. But there is a very critical point. If you cross that 150-meter line, you or your progeny has to work to come up, or you are out of the ecosystem. So this is kind of this idea called evolutionary pressure. The evolutionary pressure of density is actually very high. So it's quite strange. And it's not just diatoms. We have now, going back to many acantharians, radiolarans, which of course Haeckel discovered, are made of strontium. It's even heavier and they sink like a rock. And we have been trying, they're very rare to find. And I really admire the fact that he could discover, so many, he named 400 or so species. I mean, it was amazing to be thinking about how much time you have to spend. They're very rare to find, they're deeper waters. And then one of the big mysteries we're trying to solve is how on earth are these things climbing up when they are so heavy to begin with? There was a question here, and then I'll tell you about other part of my life. Yes?
Just out of curiosity, I wanted to know, have you compared the same species at different latitudes?
That's correct. So the types of data sets that we actually collect, and maybe, and when you say latitudes, do you mean on the planet or do you mean by depth? On the planet. So I think one of the things that is known and what you're alluding towards is as temperatures are changing, so we collect, I think we have been on maybe around 10 expeditions so far, and we will decide at what latitudes we're collecting sets of samples. We make our decisions of going out there based on satellite data. because it's very depressing to go out there, pack up 14 suitcases of all the hardware and find an empty ocean. And it happens all the time. The ocean is not just everywhere this bloom. No, it's very, very patchy. So I think it's, although I would love to do, I mean, we do, when we are out there, we do many kind of transects where we are trying to understand, but the decision of where to go is quite dependent on, and because we work on trying to couple much of this biology with climate change, we try to get right before the bloom, watch the bloom die, and watch where the carbon went. So if I had the luxury, we could do something very systematic, but that's hard. And I think this is, I will talk about the second part of that now, of how do we make much larger scale measurements on our planet possible. And that will be, I think this is a big challenge for, we are not observing at our planet at a granularity that we need. And this is where biological and scientific instruments come into play. So unless there was a burning question, I'm going to switch. How long do we go for? Do we know 12, 15? Something like that? And I'm here, so we can ask questions too.
We'll go to the Leibniz eventually. So somebody.
Okay, so let me switch to tell you about something that I really came to tell you about, which is a different part of my life, which is doing science with other people. But when I say other people, I mean people that I have never known, and people that could be in all parts of the world, all the way from, Congo to California and Nigeria to Nebraska, and do science in a community-driven context. It's hard, it's difficult, but I'll tell you why it's important. And of course, I didn't mention, I'll talk about people that are involved in the work maybe in the very end. Let me start by saying something that I recently learned from a student of mine who is in Sudan at this very moment. I teach this class called Frugal Science. It has students that join from all around the world. I met Benedict through some of those sets of interactions. It's become a large community now. And he said this to me. It's a Sudanese saying. And he said, we say this in our culture, that don't make a pen that doesn't eradicate ignorance and shame. And what he was trying to refer to is this idea that sometimes solutions really require to solve a problem. And there can be solutions, like a pen is a symbol of technology. But if a symbol of technology is something like a pen doesn't really eradicate ignorance, is it really a pen to even begin with? And I think this is a challenge that I often think about, that we create sets of technologies, but as creators of technologies, we have sometimes very little say in the societal context of how those tools will be used, or what our dreams might have been for how those tools should be used, and why it's important for us as scientists to engage in that process to really shape some of the context, or at least be participants in this societal context. And I think I'll talk a little bit about this instrument Foldscope that we have scaled at this point. But the thing that I want you to remember in some sense is all around the world at this very moment, out of the 2